Hello everyone, I'm Dr. Monica and welcome back to our tooth development series. In the previous video, we discussed the bud stage. Today, we will move on to the cap stage, one of the most important stages of odontogenesis. Now let's first understand the formation of cap. So around 9 to 10 weeks of intrauterine life, the epithelial bud, it continues to proliferate. The central cells of the bud will proliferate slower as compared to the peripheral cells. As we can see in this diagram, there is formation of a deep surface which becomes concave in shape and it resembles the shape of a cap. So at this stage, the developing tooth is termed as tooth germ. So now let's first understand the components of tooth germ. At the cap stage, the tooth germ consists of three main components. The first one is the enamel organ, which is ectodermal in origin and it gives rise to the enamel. Next is the dental papilla and it is ectomesenchymal in origin and it lies beneath the concavity of the enamel organ and it gives rise to future dentine and pulp. Next is the dental follicle or dental sac which surrounds both the dental papilla and enamel organ and it is also ectomesenchymal in origin and it gives rise to future cementum, periodontal ligament and alveolar bone. Cap stage of tooth development comprises of three cell layers. The outermost is the outer enamel epithelium, followed by stellate reticulum layer. Then the innermost is made up of inner enamel epithelium. Stratum intermedium, it is not present at the cap stage of tooth development. It appears during the early bell stage of tooth development. So coming to outer enamel epithelium, it is composed of single layer of cuboidal cells and it mainly protects the enamel organ. It maintains its shape and allows the nutrient exchange. Next is the inner enamel epithelium. It lines the concavity facing the dental papilla and it is also a single layer of cuboidal cells and it later becomes the tall columnar ameloblast and it determines the shape of the future crown. Next is the stellate reticulum. It is composed of star-shaped cells and these cells they produce the glycosaminoglycans which attract water and create spaces and the main function includes cushioning the tooth germ, protecting the inner cells, maintaining shape and allowing the nutrient diffusion. So coming to the dental papilla, it is the ectomesenchymal condensation beneath the inner enamel epithelium and it is the key site for the epithelial and mesenchymal interaction. So the dental follicle or the dental sac, it is the ectomesenchymal condensation which surrounds the enamel organ and the dental papilla and it forms the supporting structures of the tooth. So coming to enamel knot, it is called the signaling center. This is a localized cluster of non-dividing epithelial cells within the inner enamel epithelium, but it does not become part of the mature tooth. It acts as a temporary signaling center. So it releases signaling molecules such as BMP, FGF, SHS, and WNT proteins, which regulates the tooth shape, position of the future cusp, cell proliferation and differentiation, and it later undergoes apoptosis and acts like the architect of the developing crown. So another important structure formed during cap stage is the successional lamina, which is an epithelial extension of the dental lamina that appears on the lingual side of the developing tooth germ. So this successional lamina, it gives rise to the permanent successors of primary teeth, that is incisors, canines and premolars, except for the permanent molars. So talking about the significance of cap stage, whenever there is disturbed epithelial mesenchymal signaling, there will be altered enamel knot and morphogen signaling, which will lead to developmental dental anomalies affecting the tooth size, tooth shape, cusp, and the tooth number. If you have liked the content, please like, share, and do subscribe to this channel. Thank you.